Okay, so my name is Petri Auvinen, as told, so we live now in the Biocenter 2. And our lab is doing DNA sequencing and genomics, and for half of our lab is doing service, and then half of our lab is doing research on different kind of items. So this is some picture with metagenomics I want to start with. This is a tree of life made by Per Borg quite a long time ago. These are the bacteria, and these are then the metazoans, and these are the archaea. And I think this is also a kind of a picture of ourselves. So this is how we look like. So we have 100 times more, or 200 times more genes from the bacteria than our own, and about the same amount of bacterial cells in us. And as perhaps you know from the there have been, if you think about the metagenomics, in the last year or two, there have been some interesting publications about the archaea, which have now genes like in bacteria you have actin-like genes and tubulin-like genes, but in the archaea they have now found genoces which have actin and tubulin genes, and they have some other GTPases and some things which are supposed to be only in eukaryotic. And based on that data, they also have been suggesting that in fact we don't have 3 kingdoms, we have 2 kingdoms, and we are the descendants of the archaea. So all the people who work with archaea, you have to be proud of that. So this is anyway the thing that you study with the metagenome. And there are 2 ways to do it in principle. So the metagenomics, so Jenny Hultman doesn't like to say this. He hates that the amplicon stuff or targeted is called metagenomics, but that's more, I think it's still, Jenny is not here yet. It's more semantic, since with both of these you can get the same kind of in here you have a more targeted and very narrow idea, but you can nowadays use 16S, as well everybody knows, also modeling for what is the genome, kind of the population doing in the metabolism way. The only thing is that of course the accuracy is lower. Then the metagenomics that also Jenny accepts, and metagenomics is to random, so you sequence everything. So the difference between these 2 is that in here you get much deeper. You get most of the genuses and most of the organisms which are living there. And in the metagenomes, usually because of the problems of the assembly and problem of the budget, usually you can't get as deep. And if you do the targeted sequencing, so we have been doing the kind of the community stuff already with using ESTs and Sanger sequencing, and then we sequence thousands of entire 16S. And the difference between the nowadays using NGS and the 16S is that in bacteria the real stuff is in the strain level. If the shorter your amplicon is going to be, the less information you have. You have a lot of data, but there is very little of information, since you can't see which is the species there, which is the genus there. So the longer the PCR fragment you are sequencing, the more information you have. So what we are trying to do in future, we know how it would work. We use like PacBio, where you have all the sequences. So far we get too few sequences, it's too expensive to do metagenomics 16S kind of approach with using PacBio, but that's something we are planning to do. And also we are thinking to sequence the whole ribosomal operon, so 5.5 kb sequences. Then you get all the stuff which is in the bacteria and in the community. So this is just another picture of it. So most of these we have been doing in our lab. So amplicon sequencing a lot, and then shotgun genomics, and then genome assemblies. This is something that we are doing also for slightly larger genomes, like the Saimaa ring seal. This culturing we haven't been doing. So for this we have the real microbiologists working. I'm a geneticist, so I don't know too much of microbes when growing, except of E. coli. And in the metagenome aspect, you can do all of this. You can do transcriptomics, metabolomics, metabolic networks, functional assignment. All of this can be done if you have enough good data. So the data in all the analysis in metagenomics, the most important thing is what kind of a data you have and the accuracy of the data. This kind of a picture has been already seen. So for the metagenomics using 16S, one usually uses the small parts of this one, 16S gene. The variable regions, and then the variable 1 to 6 are usually okay. At least we haven't been doing V6 to V9 usually have much less information. So if you use those, the accuracy of prediction of what species you have is going to be less. So the important thing is that you have much, the more variability you have, the better it is. But this also tells you already now, if you see glance here, there is no primer which will give you in every sample every single species. There is not just, doesn't happen there. And if you think about the first picture, it's also true that there are perhaps only 100 genes which are seen in most of all of the microbes. So there are no housekeeping genes. So if you look for housekeeping genes, you can't find them even in large animals like us. So when you do this, when you select your amplicons, you will select also a subgroup of the bacteria or microbes you are going to do. ITS we have been using ITS sequencing for the fungi. It can be it's very variable. The difference between 16S and ITS is that in ITS there is a huge variance between the length. And then when there is a length problem, the length difference, then there will be some kind of a bias with the length of them. So if the amplicon is exactly about the same size, they are going to be as equally efficiently sequenced. But if you have much shorter and much longer mix, then the shorter ones are going to be faster. They just going to be sequenced more efficiently. That's just the fact of the physics on the platform. So this is by Velma, who is also here, a picture about the workflow. So what I want to show with this is there are many different phases. Some of them are in the lab, which Velma don't do, and then there's a lot of bioinformatics which he does. One of the things we have learned during there, so we have been starting doing this kind of work 2006 when we got the first the 454. It was the first of the NGS machines in Finland. We started thinking about doing the how to do the sequencing, how to use the parallel sequencing for the metagenome samples. But then in the first phase, the sequencing length was only 100 bases, and we did 16S with 100 bases, but the resolution was very low. And this comes where I will say also later, when you have 100 nucleotides of a fragment and you try to assemble a metagenome, you will fail. Since there are millions of small IS elements in bacterial genomes that you can't identify from each other. So we try to put out with the 100 bases of 454 sequences one individual bacterial genome, and you will fail. It will be scattered in small pieces. So the length matters, at least when you want to do assemblies. And then the other thing which is in here is from DNA extraction and amplification and DNA sequencing is the contamination. This is an important thing. This is something that we knew that it can be a problem, but it took a while until we get some samples that we really get problems in contamination. We have at the same time, unfortunately, 2 different projects, completely unrelated, and then we found out that there is something really wrong, that the samples, there was one set of samples we took 3 years from human skin samples. The first sample looked fine, and the second year samples were completely screwed up. And we were completely crazy in the lab and trying to find out what was the reason. The only reason we found it was something happened when the samples were taken. So most of the stuff on the samples were contamination. And then we have another sample set which have exactly the same thing. So we saw that there's something that can't be the same. So the variability of the microbes we saw was small, and then there were some of the genuses were something that seems to be that they come from something from the kit. So when your samples are very small, the contamination is a bigger problem. Since when the DNA is small, it doesn't usually work well as well. So the things coming from the lab, so there's a lab fingerprint. So every lab has their own fingerprint. And then there are contaminations with the kits. So when we do large sets of samples, we order one lot of a kit, and we test them to see the 0 samples organisms, that we know which kind of things are there. Do you really sequence what you get? It's from the sample and it's from your lab. So that's why we do it slower than we used to be doing. Since every single sample we do, it's in a hood which is sterilized by DNA, and DNA is off, and then one tube at a time opened. Not on the we don't want to automatize this, since then you get problems. If you have a small sample, small amount of DNA, you can contaminate with just a breath. Then there's the batch effect, which is something that we also found. So if you don't look for the batch effects, you don't find them. But if you look them, you will find them. And in every single thing here. Even with the DNA sequencing, you will find batch effects. And I've been seeing publications in Nature Medicine where they have one of the main founding agents was a contamination. And then there are papers where they have been publishing where the only effect which they saw was on batch effect, since the samples were coming in lab accordingly, and then they sequenced the first samples first and the late samples late, and then the only effect between the crews was that there was a batch effect because of the DNA isolation. So now we take care of, we try to get care of this by, if our own samples, when we know which samples are going to be due and how they are going to be analyzed, we try to think the whole thing before. So we mix the samples, we randomize the samples, and we randomize as possible also in the PCR amplification step. DNA sequencing, we don't want to randomize it in, of course. We want to know what the samples are, not triple blind work. But so that we sequence in all the samples which are going to be analyzed in one run. So we have small pieces of amount of reads in every run, and then run many of them to get rid of some of the problems which comes from the sequencing. Sequencing quality control, I'm not going to talk about too much. These are important parts of it. This statistical visualization, I think it's the most demanding thing, and it will take sometimes months or sometimes years to do that. So these are just a picture of Elmo, I think, as well. So the commercial the contaminants, this is something that you can't avoid. You have to know it and you have to take care of it. There are some you can use mock communities which are very good for this purpose, so they don't tell all the things, since they tell you that the sequencing works or the analysis works. It doesn't tell anything about your sample. Primary selection is very important, and then the number of cycles. So we do usually 16S with 2 cells. So first we do with short every single sample in the same time with 2 or 3 or 4 duplicates depending on the size of the project, and then minimize the cycles, and then at the second phase where then we do we add the tags for each of the samples separately. Chimeras is a problem, of course, since some of them might be true. Like if you work with viruses, there are RNA viruses which make chimeras. So if you do PCR, it might be that it's a true thing. See, like enterovirus, it's an RNA virus, they can switch the template in between. So some of the microbes also have done that during the past. So some of the 16S, the whole genes are coming from 2 different sources. But it's something that you can't avoid. Sequencing platform is something I'm going to talk about, since we have been doing quite a lot of that. Sequencing quality control. So this is from MySeq. So this is something that you have to look for. Already early on, when we did the 454 work, it was in the December 2006, and after that, what we learned is that it's better to throw away as much of data you can. You just use the best possible data. We have been doing the SIMA ring sequence. Tommy Newman also has been involved in that project. We throw away the Fred value we used is at least Fred 28 or more for all the data. When you have the so-called made-pay libraries, it was even higher, so Fred 30 or more. Since if you have a garbage data, you will get garbage out, and you can't see afterwards what happened. It's not possible to do when you have millions. So you have to trim them and take all the bad nucleotides away, and then you have to take all the bad reads away. And this is not just this is from the 454 era, but this is for whatever you sequence. So the idea is that you have the adapters, which are typical for the sequencing platform. Then you have the primers, if this is 16S, and then you have a tag, and the tag here is the important thing. That's for identification of the sample. So you can mix them, hundreds of them if you want, and then you can afterwards look for the primers. But the tag, when we started with 454, so we followed the Roche order or the instructions for one run, and then the second run we already say that, well, we are not going to do it like this, we want to do it in another way, and we started tagging before there were tags, just that we would see for if there are contamination of the samples in runs. But when to do the tags, which I thought in the beginning that it doesn't matter, it's just mathematics. You just you have 6 barcodes and you have this and this many different options. But in sequencing, it's not like that. So there are mistakes in the tag sequencing, and then the samples are going to be in the different place. So what we found in the Saimaa ring seed is a very good example for that. It's called switching. Although we have been knowing this for years, and we have been optimizing the tags for this was in Illumina, we found also the Valio GG, the probiotic bacteria genome sequence in the Saimaa ring seed, which was the option. Of course, we then decided that, okay, it takes probiotics, and it makes healthy, since Saimaa ring seems to be healthy, but it was coming from the bioinformatics contamination. So it's nothing to do with the sequencing. When you do the indexes, although they were Hamming distance from 2 different tags, which means that how many mutations in the tag you have, and until it will be mixed to the next one. Although there were 3, Hamming distance was 3, so 3 mistakes, and still it was coming from some other data set there. So this is something that we are going to change in our lab. We can't do it in one day, is to find out that how long the primates can be and have to be from each other. And then we go to the assembly. So when you do this is for the shotgun metagenomics. When you do the assembly, there are 2 ways: the de novo and the reference mapping, and they have their own problems. So if you do de novo, the longer the reads, the better is going to be the assembly. And the more accurate your data sequence, original sequence is, the more accurate and more reliable the assembly is. It's very difficult afterwards to see, if you have a large genome set, that which are the incorrect places. The reference mapping is something that in human genetics is usually used. So you have your human reference and then you map something there. They are not exclusive. So in here you see only the things that you know, and in here you can see also things which you haven't been seen before, like if you have structural variants. For metagenomics, this is exactly the same problem. So the assembly is going like this. So first you have your nice DNA, and because of the systems we have, you have to fragment it. And then you shade it, and you read it, and then you start putting it together as contigs. And then the problems comes with sequencing errors, repeats, and short reads. If you have a repeat which is 100 nucleotides in length, and you have your read length is 100 nucleotides, you can't position them. It's not possible. And that's why there have been some maths done for how long the reads should be that you can assemble one bacterial genome. And they are quite long, since they have to be much longer than the 5 kb, which is the ribosomal unit. And so if you take a human genome where the longest repeats are 7 to 8 kbs, so your sequences must be much longer so that you can identify where they are. And if there are many repeats, like the ribosomal repeats in humans, they are still not assembled. So there are holes, you can't do that, even with a backbio. And this is the problem. I will just shortly go through this. So how you do it, how you assemble, you have this kind of graphs. So some of my people wrote them by pencil in the papers, since it helps to understand how they are. So what you want to know is what is the way that you can assemble and have the different parts in the correct order, if you don't know it before. So this is a mathematical problem. And why the repeats are problems, this is the reason for that. So these are the repeats, and you don't know exactly what is the road here. So when you have a metagenome, you can have even more of this kind of a problem. So you have to be critical of how you do the assembly. This is difficult even if you had one genome. And this is what I like. This is by Panu Somervo, the picture. This was the time when we're doing the butterfly genome together. So longer reads are better than the short reads if you want to assemble them. That's the same thing with metagenomes. But usually the longer reads are more expensive than the short reads, and that's why even we do also the short reads, and then we have a battle of what kind of a data is good and which is bad when you have you can't see the reference since it's not existing. This is how they look like. When you have the metagenomes, I don't know how complicated. We haven't been visualizing them, but even one genome, this is how it looks like. So these are just some bacterial genomes which are put together. And then we go to the technology. So this is the Illumina, how it works. So it was first called Solexa. It was an English-based company, and then I have been in the meetings where they announced that now we can sequence 3 nucleotides, I mean out of the 4. And then the next year they say that, okay, now we can sequence all the 4 nucleotides. And the read length was 20 nucleotides. And everybody were heroing, this is fantastic. And they do sequenced the SNP published in Science, a paper about human SNPs by sequencing 20 nucleotides of length. Now we sequence a little bit more. In all the systems with the next gen, the idea is that you have a library, which is your sample. It's an amplicon or whatever, and then you have two different kind of oligos which are needed for the sequencing platform to perform the sequencing. In Illumina, you have a class where there is randomly distributed oligos on the template, which are complementary to these ends of the fragments. You dilute the sample so that the individual molecules are not adjacent. They have to be enough, depending on the sequencing machine, they have to be enough long from each other. And then you start doing PCR, called bridge PCR, on the slide. And the end, you will end up with this kind of a cluster. So first you have a bridge which goes like this, and then the machine, you don't have to do pipetting here. It's the machine is doing, fortunately, everything. And then you open it, then you have a linear DNA which is as a cluster. And they are very near each other. This is just a picture of that. So we have millions or hundreds of millions per small, or thousands of them in 1 square millimeter. So this is now from one individual molecule. And as you can imagine, if you have too much of DNA there, you can have two molecules in the same context. And then that we are usually seeing in the sequencing, they are discarded. So that's why it's really picky. And some of the machines are unbelievably picky how much of DNA you put there. So some of the libraries work better and some of them don't. The sequencing is like Sanger in the Illumina case. So you have your cluster here, you strip off one of the strands, you add a primer, and then you start adding one nucleotide at a time, like in Sanger sequencing. So the polymerase can add one nucleotide, then the machine takes a picture. Then it cleaves off the color, you add the next nucleotide, and then you read that. And then in this case, then they identify the clusters. In real life, the clusters are just adjacent to each other. Depending on the machine, the size of how many molecules you have there and how large the cluster is depends on the optics of the machine. And then you go on with this as many times that you you need to do that. And these are most of not all the Illumina machines. One thing you have to keep in mind, all the machines, there is no one box which is suitable for all the projects. There is not, some people think, but it's not probable. So we have two of the MySeq and NextSeq, and then we have BacBio, which we're going to talk about later. So most of the amplicons are done with the MySeq, since you have the longer read length. So it's 2 times 300 bases. So paired end, one end, and then the other end. The problem with Illumina has been, we have also seen that for some reason the 300 bases, the read length, it has gone, quality has gone down. So we have to be doing some of the sequences, repeating the amplicons and changing the amplicons, since it didn't anymore read so that we have good enough sequences to overlap them. And that's something we were. The representative of Illumina in Finland doesn't want to talk with me, so he usually likes to talk with Lasse and Pia, since I don't like that it doesn't work. So, but there is nothing that they can do. There's something in the chemistry that they can't correct. This has been we have been complaining this, and also other people. First, they said to us that nobody has the same problem, but we know that we have the problem. So you can get something like 20 million paired end reads in here. This we use also when you resequence hundreds or sometimes hundreds of microbe genomes. This is something that, since the longer you have, the more easier it's going to be. Next is the monster already. So the read length is shorter. It's 2 times 150 base pairs. But you get in 800 million paired end reads, and it takes 1 day. So this gives you human genome 30 times in 24 hours or something like that. So we have using this machine that sequenced 200 seal genomes in a lower coverage. This is for the metagenome if you have a shotgun. But in here the problem comes that what size of a library is it, and how accurate the sequence is going to be. The HiSeq is something that is used mostly for human genomes or RNA-seq. This is also very for RNA-seq. The price per base is about the same with these. It's kind of a different applications what you do with those. RNA-seq with this machine, you get 400 million reads in 10 hours. So the library making and the analysis takes much longer than the run. So. And then we have the ion proton. I know that Anu has been talking about this. This is something that we haven't been using at all, and I show you soon why we don't use it. So the proton is something that they promised first of all the chip 2 for years, and they failed to do that. And I show you soon why they did fail to do that. As such, it's an intelligent system. So if you remember 454, that's not anybody. It's the 454 we have is in the University Museum now. The box for there. So who remembers how DNA polymers work? Nah, come on. So when in the 3' end, when there is the polymerase adds a new nucleotide, there are two things which are then released. There's a pyrophosphate, and then there's a proton. And in the 454, they use the proton, the pyrophosphate to create light. So they have an enzymatic cascade of 4 different enzymes, and then they get the light we have a CCD camera measuring the light intensity there. And the problem there is it's an enzymatic reaction, so it's not like in Illumina where you just read as long you want, but there's a peak for the enzymatic reaction. That's a problem with the 454, and which has more problems in homopolymers, that you can't, when are you going to detect it? It is this many minutes or this many seconds afterwards that you can accurately measure it. So the guy who invented the 454 machine also invented this one, and he then in the ion they use the proton which is released there. So it's a small pH meter, and in both 454 and in the ion, the idea is that you don't have a glass plate, you have a bead, and you dilute your DNA with the suitable ends there on the bead, put it in the lipid droplet like a mayonnaise, and then you make the PCR out of there. And that's the way you clone your DNA, since you need to have many copies of the same molecules in case that you can detect it. So the proton chip 2 failed, that the beads were getting so small that there was a leakage between the wells. They couldn't anymore see it reliably. That was the bad thing for the proton, since that would be the only machine that would give enough data compared to the Illumina machines. So it's a good idea, but it's since it used non-nucleotides which are normal, so they are very cheap to use. But that's the accuracy is the problem, since you measure the electric voltage. Then we go to the PacBio. So this is then the science fiction part. So this is then I don't know whether I know that some of you know it. So this is single molecule sequencing. So in all the other systems, so-called second generation sequencing systems, you amplify one single molecule which you sequence just to increase the signal. But in here you have one molecule. So you have a smart cell, as they say. You have all the nucleotides, as in Sanger or Illumina, they have their own color. And the only thing here is that it has 6 phosphates. I don't know whether it's because of some patents or whatever, but they have 6 phosphates instead of 3. You have a small hole, 100 nanometers in diameter, and then you have 2 lasers in the bottom of the machine is huge. It's like I think 1,000 kilos, whatever. It's an amazing machine, and it's size of my fingertip is where the sequencing happens. But it's 100 nanometers in diameter, and there are 2 lasers, green and red laser, in the bottom of that, and then they illuminate all these 150,000 small holes which are in the bottom of this machine, of this smart cell. And the idea for that is that, as you can imagine, in a tube there are all these nucleotides. If you would inlight with laser the whole tube, it will be completely illuminated, and you couldn't see the signal. Since in the bottom of here there is one polymerase with one DNA molecule there. And the idea, who of you knows the way the microwave, what is the length of a microwave in a microwave oven in a home? It's 10 centimeters. And that's why the holes are small. Since if you would go home and you would cut in between the holes, so you have a 10 centimeter length, you will boil yourself. And that's the idea why you can see in the oven. And in here is exactly the same idea. So the wavelength of the lasers is from 400 to 650 nanometers. So the light doesn't enter the hole and doesn't illuminate the whole tube. It illuminates only 20 ceptoliters on here. So it's just a small amount of area around the polymerase is illuminated, and that will then, when the nucleotides come in the polymerase, it in vivo all the time reads 70 times per second. It reads the video is taken there, and then it's sequence. So then you have a signal out of there. So this goes on and on. So that's how it looks like. It's called Smart Bell. So this is your template, and then we add the oligonucleotides where there is a primer attachment and then a polymerase. And this is then, it's not pipetted there, as you can imagine. It's just put it in, there's only billion molecules which are used in one smart cell to sequence. And then it starts reading, and it goes as long the polymerase wants to go. And then it drops off. And then this is the same polymer, so this is first here, then it comes here, and then comes here, and then it goes around there, depending. If you have very short, it can go many, many times. Then these are the subreads, so then you take off these parts, and then you can assemble them, and then you have a consensus called read of insert. So this is used in putting together, has been used for putting together metagenomes. We haven't been using it. It's fairly expensive, but you can get much further with that. The problem with the single molecule sequence is that the accuracy is only 87%. So, but the good thing is that it's mostly random. So when your coverage is higher, the accuracy will be higher. After 20x, the higher accuracy will be higher than in any Illumina machine ever, since there is a problems with Illumina making systemic errors which you can't filter out. So we have been doing this for some viruses which are 5 kbs, and we get accuracy. So the Q value is something with 50 or 60 when you do it many, many times. And the other good thing with PacBio compared to all the second generation sequencers with Amplify is that it's the coverage for different contents of GCs about equal. This is exactly what you want. Since if you have a high or low AT or GC, you usually with Illumina whatever machine, you will lose them because of problems. But in here, it's going to be easier. Homopolymers are problems for everything, even with Sanger. It just depends how long it's. In 454, when you have, in a certain context, when you have after 3 or after 4, you get problems. What is the accuracy there? And it depends in here in PacBio as well. So that's why it is something that you have to, in metagenomes, you have to be more accurate with those. With the PacBio, one of the additional good things, what we have been using in our advantage is that when you have small genomes or bacteria genomes, when we put it together, you can also get the methylome out of that. So you can get the massively analyze all the methylation patterns, and even some of the what methylations are in the bacteria. So that's epigenetics of bacteria we have been also working for with our own projects. Then the science fiction part is this is also single molecule. This has not been very accurately working so far. This is the MinION from Oxford Nanopore, where you have a nanopore on an artificial plasma membrane, and then when it goes through, it reads 5 nucleotides at a time. So there are 1,024 different forms of all the 5 nucleotides, and then you have a matrix which tells you this is the sequencing result. So this is the machine. I really don't understand what it means. Usually I understand what the sequencing comes out of there, but in here I don't have a faintest idea. We have the MinION, but last year when we moved, we didn't have time to do anything, so we are going to try this as well. So there have been some reports of the version 9 these pores that they seems to be working fairly well. So that beginning I know it 2 years ago I was in Embl and talked with the genome unit director there, Vladimir Benes, and he was using MinION and sequence lambda phage. And then I just asked him, we didn't have it then, and how did it work? And they, well, we put lambda phage in, and when we get the sequence out, they blasted it, and they couldn't find lambda. So, and that tells you how. So now it's getting better. So I think in a short while, these are going to be useful for something. The problem here for 16S is that since you have many indels, and so the accuracy is going to be probably not, at least now, suitable for 16S. Since if you have one nucleotide difference, it's a different bug. But they have been doing like the Ebola, you remember the Ebola outbreak. So they were sequencing in the forest using this one, identifying the Ebola viruses there. Since you can use it really with the laptop. So you have a USB, you put the laptop, it's the size of your smartphone. So it is coming, I think, this in future. We hope that we have time to test that. One of the things we have to, with anything you do, what any science you do, is that you have to think about which is suitable for your assay. So when you are going to do, this is just a short list of the machines, you have to choose. So when you do your experiment, you have to think about the analysis already. So I have been telling to many of my collaborators that please discuss with us before you do the experiment, so that you can think about how the analysis is going to be. Probably it's going to be changed during the project, but anyway, try to do, like when you do clinical samples, that you have enough samples. So we have been seeing, we have a difference between the medical doctors and us. What is an individual sample? So when people say that you have this many samples and you find out that only 1/3 of them are suitable for the analysis, and then the N is too small. So whatever you do, choose it before and discuss with people who sequence and the people who analyze the data prior you do anything. Since I have been also, when I was doing more than 10 years ago microarray analysis, and sometimes we get some people coming in the lab, could you help us to analyze? And then we saw the data, well, you can't do anything with this, since already the experiment has been misdesigned. So you couldn't do any analysis of the data. And people throw away thousands of euros. And then for the analysis part, this is just one example with it was Jenny and then Kaisa, who is now in Austria. This is the only thing I want to, and it's reminding for me, is that when you do the analysis, there is no solution that when you push the button, you get really the correct answer. In whatever you do, you have to know your tools. So even if you like Mother or you like Chime, whatever you do, but understand the algorithms behind that, and whether the results make any sense. That was told me when I was in high school in physics lessons, that when you do, you have to have it, you have to make the calculation in your head first, that you know that when the results, when it comes out, does it make any sense? When you do the analysis, this is just how we. So I have a simple question for Kaisa and Jenny. I wanted to. It was so simple in the beginning, that how should we analyze the 16S data? So what methods and how should we use the parameters? And the problem was that it took 1.5 years to find it out for these two ladies, how to do that. And the end result was astonishing, since there were some of the systems were creating like 1,000 times more OTUs. Since if you didn't understand how the algorithm works, it created OTUs from the data. This was 454 times, so there was. So that's you have to do be careful for. For the assemblies, I can tell you one sorrow story, which was from the SIMA ring seal, which we haven't. So we were doing the SIMA ring seal assembly, which it's 2.5 gigs. It's a huge genome to put it together, and it was going well, and all the numbers were looking suitable. But then we did also the gray seal, and then we did the same methods with the gray seal, and unfortunately then everything collapsed, and we have to go backwards because there was something wrong with the assembly. And we have to go 2 months back in the SIMA ring seal and redo everything, since one program which we thought it looked okay, but it was not. So when my student Juhana Kammonen was looking through the whole code, then he found out the reason why it was doing mistakes. So know your books and ask somebody. And I think that's the end for, and you get coffee.
Actually, is this on? Yeah. So actually, just a comment. So the latest comparison between Ion PGM and MySec data that was done. Actually, we got the results last week. So the results from those are already online. The slides from my yesterday lecture. But yeah, I agree. PGM data is never better than MySec data. But I do say it is more affordable.
It is so. I saw the prices. So you compare the prices. I think they are not. They are orange. No. Yeah. The €7 and €50, they are completely different things. The €50 have everything before the sequencing also. So we did everything. So that's not accurate for that. So if you do metagenome, so Well, I know that in Jyväskylä, I know that there's a group who has been doing a microbe genome, put it together with MySic data, and it took 2 years, and they couldn't put it together because of the mistakes. Since when you do the assembly, you have SNPs in there. The assembly, you have to have a parameters how many SNPs are allowed when you put it together, and it was not ever done. So then we did it with the PacBio. It took 4 weeks to put it together. So when you do metagenome assembly, the quality of data you use is even more important, since you can't. It's very difficult to see afterwards whether the linkage is correct or not.